ਇੱਕ ਨਿਊਕਲੀਅਸ ਵਿੱਚ ਪਾਏ ਜਾਂਦੇ ਹਨ, ਪਰ ਪ੍ਰੋਕੈਰੀਓਟਸ ਵਿੱਚ DNA ਸਾਈਟੋਪਲਾਜ਼ਮ ਵਿੱਚ ਫਲੋਟ ਕਰਦਾ ਹੈ, ਜਿਸ ਨੂੰ ਨਿਊਕਲੀਓਇਡ ਕਹਿੰਦੇ ਹਨ। Now we come to the end of today's class. Let us revise what we have studied. First of all, we discussed about Golgi apparatus, also known as Golgi bodies or Golgi complex. It was identified in 1898 by the Italian physician Camillo Golgi and was named after him. The primary function of the Golgi apparatus is to process and package macromolecules such as proteins and lipids after their synthesis and before they make their way to their destination. Lysosomes are spherical organelles that contain enzymes. They are found in animal cells, while in yeast and plants, the same roles are performed by lytic vacuoles. Lysosomes digest excess or worn-out organelles, food particles, and engulfed viruses or bacteria. Next was ribosomes. Ribosomes are cytoplasmic granules composed of RNA and protein, at which protein synthesis takes place. They were first observed by Palade in the electron microscope as dense particles or granules. Each ribosome is composed of two subunits. One is larger in size than the other. Ribosomes are non-membrane bounded particles where protein synthesis occurs. Next, we talked about centrosome, which contains two barrel-shaped structures known as centrioles. The walls of each centriole are usually composed of 9 triplets of microtubules. As mitosis proceeds, thread-like structures grow from centrosome with their movement towards the metaphase plate. These clusters of microtubules are called spindle fibers. We also discussed the control center of a cell, cell nucleus, which contains the cell's chromosomal DNA. DNA is wrapped around the protein histones and then twisted together again in the shape of a spring. Types of chromosomes were also discussed with you, which included the metacentric chromosome, which has the middle centromere forming two equal arms of the chromosome. Then the submetacentric chromosome having centromere slightly away from the middle of the chromosome, forming one shorter arm and the other longer arm. There were two more types, acrocentric and telocentric. In case of the acrocentric chromosome, the centromere is situated close to its end, forming one extremely short arm and a very long arm, whereas the telocentric chromosome has a terminal centromere. चलो हुण इन्ना सब जान लैन दे बाद हुण चेक करिए कि तुहानू किन्ना कुछ समझ आया है। सब तो पहला गोल्जी बॉडी बारे पूछ लईए। So the question is, describe the structure of Golgi apparatus. The answer is, the Golgi apparatus comprises a series of 5 to 8 cup-shaped membrane-covered sacs called cisternae that look something like a stack of deflated balloons. My next question is, how do lysosomes perform the function of digestion? The answer is, to accomplish the tasks associated with digestion, the lysosomes use some different types of hydrolytic enzymes present in them. This is a picture of the nucleus. Identify nuclear membrane, chromatin, and perinuclear space in this figure. Answer: These thread-like structures form chromatin. It is bounded by a double membranous layer, nuclear membrane. The space between these two layers is known as perinuclear space. The next question is, identify the type of chromosomes given here in the picture. Answer. This is submetacentric and this one is telocentric type of chromosomes. I wish I could ask you more questions, but the time for today's class is over. I am confident that you have learnt enough about these some of the very important organelles. Looking forward for the next class. Thanks for your attention and see you next time. Cell, the unit of life. ਬੱਚਿਓ, ਅੱਜ ਦੀ ਕਲਾਸ ਵਿੱਚ ਤੁਹਾਡਾ ਸਵਾਗਤ ਹੈ। ਅੱਜ ਅਸੀਂ ਸੈੱਲ ਦੇ ਕੁਝ ਮਹੱਤਵਪੂਰਨ ਆਰਗੇਨੇਲੀਜ਼ ਬਾਰੇ ਗੱਲ ਕਰਾਂਗੇ, ਜਿਵੇਂ ਕਿ ਗੋਲਜੀ ਬੋਡੀਜ਼, ਲਾਈਸੋਸੋਮਸ, ਵੈਕਿਊਲਸ, ਰਾਈਬੋਸੋਮਸ, ਸੈਂਟਰੀਓਲ ਅਤੇ ਨਿਊਕਲੀਅਸ। ਅਸੀਂ ਇਹਨਾਂ ਦੇ ਬਾਰੇ ਕੀ ਜਾਣਾਂਗੇ, ਇਹ ਤੁਸੀਂ ਨੋਟ ਕਰ ਲਵੋ। Structural and functional aspects of Golgi bodies. इसी तरह structural and functional aspects of ribosomes. इस सब दे नाल ਇਹ ਵੀ ਜਾਣਾਂਗੇ ਕਿ ਸੈੱਲ ਵਿੱਚ lysosomes ਅਤੇ vacuoles ਦਾ ਕੀ ਰੋਲ ਹੈ। Role of lysosomes and vacuoles in a cell. ਅਸੀਂ ਇਹ ਵੀ ਜਾਣਾਂਗੇ ਕਿ centriole ਸੈੱਲ ਵਿੱਚ ਕੀ ਕਰਦਾ ਹੈ ਅਤੇ ਇਹ ਕਿੱਥੇ ਹੁੰਦਾ ਹੈ। ਭਾਵ structural and functional aspects of centriole. ਇੱਕ ਬਹੁਤ ਮਹੱਤਵਪੂਰਨ organelle ਹੈ nucleus, ਜੋ ਕਿ ਕਿਸੇ ਵੀ यूकेरियोटिक ਸੈੱਲ ਦੇ ਅੰਦਰ ਈਜ਼ੀਲੀ ਵੇਖਿਆ ਜਾ ਸਕਦਾ ਹੈ। Structural and functional aspect of nucleus. ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਸਾਰੇ ਜਾਣਦੇ ਹੋ ਕਿ all living organisms on earth are composed of billions of cells. Cells include different organelles. ਇਹ different organelles ਵੱਖ-ਵੱਖ ਤਰ੍ਹਾਂ ਦੇ ਕੰਮ ਕਰਦੇ ਹਨ ਅਤੇ ਸੈੱਲ ਨੂੰ ਸੁਚਾਰੂ ਢੰਗ ਨਾਲ ਚਲਾਉਣ ਵਿੱਚ ਮਦਦ ਕਰਦੇ ਹਨ। ਬਹੁਤ ਸਾਰੇ ਸੈੱਲਸ ਮਿਲ ਕੇ larger organization tissues ਬਣਾਉਂਦੇ ਹਨ ਅਤੇ ਇਹ ਟਿਸ਼ੂਜ਼ ਮਿਲ ਕੇ ਸਰੀਰ ਦੇ ਸਾਰੇ ਸਿਸਟਮਸ ਬਣਾਉਂਦੇ ਹਨ। ਸੋ ਸੈੱਲ ਬੇਸਿਕ ਯੂਨਿਟ ਆਫ ਲਾਈਫ ਹੁੰਦੇ ਹਨ ਔਰ cells are small compartments that hold all the biological organelles necessary to keep an organism alive and successful on earth. ਚਲੋ ਹੁਣ ਅੱਜ ਦੇ ਟੌਪਿਕ ਨੂੰ ਸ਼ੁਰੂ ਕਰੀਏ। ਸਭ ਤੋਂ ਪਹਿਲਾਂ ਗੋਲਜੀ ਬੋਡੀ ਬਾਰੇ ਗੱਲ ਕਰਦੇ ਹਾਂ। ਗੋਲਜੀ ਬੋਡੀਜ਼ ਨੂੰ ਗੋਲਜੀ ਐਪਰੇਟਸ ਵੀ ਕਿਹਾ ਜਾਂਦਾ ਹੈ। ਪਹਿਲਾਂ ਇਸ ਦੇ ਕੁਝ ਵਿਜ਼ੂਅਲਸ ਦੇਖ ਕੇ ਇਸ ਨੂੰ ਸਮਝਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰੋ।
Golgi apparatus. This organelle is found in almost every cell. It is named after the Italian histologist Camillo Golgi, as he was the first to observe a densely stained reticular structure near the nucleus. He named it as Golgi apparatus. The Golgi apparatus typically comprises of a series of 5 to 8 cup-shaped membrane-covered sacs called cisternae that look something like a stack of deflated balloons. The Golgi apparatus principally performs the function of packaging and transporting of materials, which is secreted inside the cell but needed outside the cell. Material to be packaged is received from the endoplasmic reticulum. This material, received from endoplasmic reticulum, fuses from one side of the Golgi apparatus and moves towards the other side. This is the reason that Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes present on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase.
ਜਿਵੇਂ ਕਿ ਤੁਸੀਂ ਵੇਖ ਰਹੇ ਹੋ, ਗੋਲਜੀ ਐਪਰੇਟਸ ਵਿੱਚ ਸਿਸਟਨੇ ਇੱਕ ਦੇ ਉੱਪਰ ਇੱਕ ਪਏ ਹੋਏ ਹਨ। ਇਹਨਾਂ ਦਾ ਇਹ ਵਾਲਾ ਕਨਵੈਕਸ ਫੇਸ ਔਰ ਫਾਰਮਿੰਗ ਫੇਸ ਹੈ, ਜਿਸ ਨੂੰ ਸਿਸ ਕਹਿ ਸਕਦੇ ਹਾਂ, ਅਤੇ ਇਹ ਦੂਜੇ ਪਾਸੇ ਕੌਨਕੇਵ ਫੇਸ ਔਰ ਮੈਚੁਰਿੰਗ ਫੇਸ ਹੈ, ਜਿਸ ਨੂੰ ਕਿ ਟਰਾਂਸ ਕਹਿ ਸਕਦੇ ਹਾਂ। ਹਰ ਆਰਗਨਲ ਦਾ ਸਿਸ ਅਤੇ ਟਰਾਂਸ ਫੇਸ ਵੱਖਰਾ ਪਰ ਇੰਟਰਕਨੈਕਟਡ ਹੁੰਦਾ ਹੈ।
ਲਾਈਸੋਸੋਮ ਗੋਲਜੀ ਐਪਰੇਟਸ ਨਾਲ ਰਿਲੇਟਡ ਇੱਕ ਹੋਰ ਸੰਰਚਨਾ ਸੈੱਲ ਵਿੱਚ ਪਾਈ ਜਾਂਦੀ ਹੈ, ਜਿਸ ਨੂੰ ਅਸੀਂ ਲਾਈਸੋਸੋਮਸ ਕਹਿੰਦੇ ਹਾਂ। ਇਹ ਲਾਈਸੋਸੋਮਸ ਸੈੱਲਸ ਦੇ ਗਾਰਬੇਜ ਡਿਸਪੋਜ਼ਲ ਸਿਸਟਮ ਵਜੋਂ ਕੰਮ ਕਰਦਾ ਹੈ। ਕ੍ਰਿਸਚੀਅਨ ਡੇ ਡੂਵੇ ਨਾਂ ਦੇ ਸਾਇੰਟਿਸਟ ਨੇ 1955 ਵਿੱਚ ਲਾਈਸੋਸੋਮਸ ਨੂੰ ਡਿਸਕਵਰ ਕੀਤਾ ਸੀ। The lysosome is a single membrane bag. It encloses a dense stroma and a large vacuole. The vacuole contains hydrolytic enzymes that are used to digest macromolecules. The lysosome contains over 40 enzymes, most of which are hydrolytic enzymes, some of which are the proteases, nucleases and lipases. ਹੁਣ ਤੁਸੀਂ ਸੋਚੋਗੇ ਕਿ ਗੋਲਜੀ ਐਪਰੇਟਸ ਅਤੇ ਲਾਈਸੋਸੋਮਸ ਦਾ ਕੀ ਰਿਲੇਸ਼ਨ ਹੈ। In fact, lysosomes are membrane-bound vesicular structures formed during the process of packaging by the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of enzymes. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. They may digest worn-out organelles such as mitochondria. Lysosomes are rarely present in plant cells but are very common in animal cells.
Lysosomes. Shape and structure of lysosomes vary with the state of the cell and its degree of activity. Lysosomes have pieces of membranes, vacuoles, granules, and parts of mitochondria inside them. To accomplish the tasks associated with digestion, the lysosomes use some different types of hydrolytic enzymes, all of which are manufactured in the endoplasmic reticulum and modified in the Golgi apparatus. Lysosomes are often pinched off from the membrane of the Golgi apparatus.
ਹੁਣ ਅਸੀਂ ਗੱਲ ਕਰਾਂਗੇ ਵੈਕਿਊਲ ਦੇ ਬਾਰੇ। ਵੈਕਿਊਲ ਦਾ ਮਤਲਬ ਹੈ empty space, ਭਾਵ ਖਾਲੀ ਥਾਂ। ਇੱਕ ਸੈੱਲ ਦੇ ਅੰਦਰ ਬਹੁਤ ਸਾਰੇ ਵੈਕਿਊਲਸ ਮਿਲਦੇ ਹਨ। But the vacuole is membrane bound space found in the cytoplasm. These were discovered by Perner. Vacuoles are storage bubbles found in cells. They are found in both animal and plant cells, but are much larger in plant cells. Vacuoles might store food or any nutrient a cell needs to survive. They can even store waste products, so the rest of the cell is protected from contamination. These are bound by a single membrane. This membrane is known as tonoplast. This is a very active and dynamic membrane. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. Young plant cells often contain many small vacuoles, but as these cells mature, these unite to form a large central vacuole. Let us see how they look like in plant cell as well as in animal cells.
Vacuoles. In plants, vacuole usually occupies most of the space in a cell. When a plant cell has stopped growing, there is usually one very large vacuole. Sometimes that vacuole can take up more than half of the cell's volume. The vacuole holds large amounts of water or food. Tonoplast means covering membrane of vacuole that facilitates the transport of a number of ions and other materials. The size of a cell may still increase or decrease depending on how much water is present in its vacuole. In animal cells, they are smaller in size and many in number. In amoeba, the contractile vacuole is important for excretion.
Ribosomes were first observed in animal cells by George Palade under an electron microscope. He found them as dense and granular particles. In plant cells, they were first of all observed by Robinson and Brown. As far as their occurrence and distribution is concerned, they are found in all cells. In prokaryotic cells, ribosomes often occur freely in the cytoplasm. In eukaryotic cells, ribosomes occur freely as well as attached to endoplasmic reticulum. These ribosomes are composed of ribonucleic acid, that is RNA, and proteins, and are not surrounded by any membrane. The eukaryotic ribosomes are ATS type, while the prokaryotic ribosomes are 70S type. Here S, that is Swedberg's unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70S and 80S ribosomes are composed of two subunits. Let us see the structure of a single ribosome.
Ribosome. Each ribosome is composed of two subunits. One is larger in size than the other. The two ribosomal subunits remain united with each other due to specific concentration of magnesium ions. When concentration of magnesium ions goes below a critical level, both subunits get separated. Similarly, by raising magnesium concentration, two subunits become associated and the structure is known as dimer. Ribosomes may occur simply as monosomes, and there is another type of association which is formed by many ribosomes forming a chain on a common messenger RNA and is called polysomes. Chemically, ribosomes are complex, bead-like structures composed of about 40% protein and 60% ribosomal RNA. Ribosomes may number millions in one cell.
Functions of ribosomes. Structure ਦੇ ਬਾਅਦ ਹੁਣ ਜੇ ribosomes ਦੇ functions ਦੇ ਬਾਰੇ ਗੱਲ ਕਰੀਏ ਤਾਂ ਇਹਨਾਂ ਦਾ main ਕੰਮ protein ਦਾ synthesis ਕਰਨਾ ਹੁੰਦਾ ਹੈ. Means the ribosomes play a very important role in protein synthesis, which is the process by which proteins are made from individual amino acids. Without the ribosomes, the message would not be read. Thus, proteins could not be produced. So, ribosomes are popularly known as protein factories. इसके अलावा ribosomes also furnish enzymes and factors needed for the formation of polypeptides and other functions. Therefore, ribosomes play a very important role as cell organelles. ਹੁਣ ਅਸੀਂ ਗੱਲ ਕਰਦੇ ਹਾਂ ਦੋ ਹੋਰ ਬਹੁਤ ਹੀ ਮਹੱਤਵਪੂਰਨ cell organelle, centrosome and centrioles ਦੀ. Centrioles are contained within a structure called centrosome. ਅਸੀਂ ਕਹਿ ਸਕਦੇ ਹਾਂ कि centrosome is an organelle which contains two cylindrical structures called centrioles. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. आओ इन्हां ਨੂੰ ਵੇਖ ਕੇ ਇਨ੍ਹਾਂ ਬਾਰੇ ਹੋਰ ਜਾਣਨ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰੀਏ.
Centriole. The walls of each centriole are usually composed of 9 evenly spaced peripheral fibrils of tubulin protein. Each of this fibril is a triplet. If we see their connectivity, each of these triplet is linked to next one. The central part of the centriole is also proteinaceous and is called hub. This hub is connected to the tubules by radial spokes. These spokes are also made up of proteins.
Function performed by centrioles. Centrioles form the basal body of cilia and flagella. Acentrioles cell division ਦੇ ਦੌਰਾਨ spindle ਵੀ ਬਣਾਉਂਦੇ ਹਨ. Spindle ਬਣਾਉਣ ਲਈ they duplicate during S phase of the cell cycle. Just before mitosis, the two centrosomes move apart until they are on opposite sides of the nucleus. As mitosis proceeds, microtubules grow out from each centrosome toward the metaphase plate. These clusters of microtubules are called spindle fibers. चलो हुण असी उस ऑर्गेनेली बारे गल करिए जो सब तो पहला सेल विच पछाणी गई सी। ओ है न्यूक्लियस। एंटोनी वैन ल्यूवेनहॉक ऑब्जर्व्ड अ ल्यूमेन पाव अंदरूनी खाली था in the red blood cells, which was later identified and described as the nucleus by Robert Brown. हुण तुसी सोचोगे कि RBC विच ते न्यूक्लियस हुंदा ही नहीं, पर ओ मनुख दी RBC नहीं सी, ओ salmon fish दी RBCs सी. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. चलो हुण इस दे विजुअल्स नू वेख के इस नू समझण दी कोशिश करिए.
Nucleus. Electron microscopy has revealed that the nuclear envelope consists of two parallel membranes with a space in between called perinuclear space. Nuclear envelope or nuclear membrane is a double-layered membrane having pores in it. The membrane separates the content of nucleus from the cytoplasm. Nuclear pores. These are the pores present in the nuclear membrane. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. Chromatin are thread-like structure composed of DNA and some basic proteins called histones. Chromatin condenses to form chromosomes during cell division.
Normally, there is only one nucleus per cell, but recently एक तो ज़्यादा nuclei भी वेखे गए ਹਨ. कुछ cells ਤਾਂ ਇਹੋ ਜਿਹੇ ਹਨ ਜਿਨ੍ਹਾਂ ਦੇ ਅੰਦਰ nucleus ਹੁੰਦਾ ਹੀ ਨਹੀਂ. Means some mature cells lack nucleus. Example, erythrocytes of many mammals and sieve tubes of vascular plants. चलो हुण सेल दे अंदर प्रेजेंट न्यूक्लियर मटीरियल बारे होर गल करिए. जिवें कि असी वेख्या कि थ्रेड लाइक स्ट्रक्चर जिसनू असी क्रोमैटिन आखदे हां न्यूक्लियस विच हुंदा है. This chromatin condenses to form chromosomes during cell division. A single human cell has approximately 2-meter long thread which get condensed to form its 46 chromosomes, means 23 pairs. Let us see the structure and shapes of chromosomes.
A chromosome is composed of two identical chromatids attached by a centromere. Centromere divides the body of chromosomes into two parts. Usually, one is shorter than the other. The DNA is wrapped around the protein and then twisted together again in the shape of a spring. Each chromatid is made up of tightly bound DNA wrapped around histones. The centromere is basically a small constriction on the sides of which disc-shaped structures called kinetochores are present. Based on the position of the centromere, the chromosomes can be classified into 4 types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of the acrocentric chromosome, the centromere is situated close to its end, forming one extremely short arm and a very long arm, whereas the telocentric chromosome has a terminal centromere.
Significance of chromosomes. चलो हुन chromosomes दी importance बारे गल करिए. Students, chromosomes are things that make organisms what they are. They carry all of the information used to help a cell grow, thrive and reproduce. ਇਸ ਦਾ ਕੀ ਕਾਰਨ ਹੈ? ਅਸੀਂ ਵੇਖਿਆ ਕਿ chromosomes DNA ਦੇ ਬਣੇ ਹੁੰਦੇ ਹਨ. Genes ਅਤੇ DNA ਦਾ ਆਪਣਾ relation ਹੈ. Segments of DNA in specific patterns are called genes. Your genes make you who you are. ਤੁਹਾਡੇ ਅੰਦਰ ਅਤੇ ਸਾਰੇ eukaryotes ਦੇ ਅੰਦਰ chromosomes